Some people hate it. Some thinks it's the greatest discovery of 19th century. Of course, we are talking about the glycolysis. Let's start with what is glycolysis. To put it very simply, glycolysis is the oxidation of glucose into pyruvate or lactic acid. Glycolysis occurs in every living cell in both aerobic and anaerobic conditions, and it occurs inside the cytoplasm. In the cytoplasm, the glucose gets converted into pyruvate, and then depending on the presence of oxygen, the pyruvate will either be converted into acetyl-CoA or it will be converted into lactate. However, this part where the glucose gets converted into pyruvate is known as glycolysis. It all starts with a glucose molecule in the cytoplasm. Now the glucose, we know it's a 6-carbon compound. A phosphate molecule attaches to this glucose molecule, and thus it becomes glucose 6-phosphate. The glucose 6-phosphate then converts into fructose 6-phosphate. It is chemically similar, but the structure gets a bit twisted. And then this fructose 6-phosphate binds with another phosphate molecule and forms fructose 1,6-bisphosphate. Now, if you notice closely, what happens here is that two phosphate molecule joins, one at the position of carbon 1 atom and the other one at the position of carbon 6 atom. Now, this makes this molecule unstable, and so it breaks up in the center, forming 3 carbon compounds. One of them is the glyceraldehyde 3-phosphate with 3 carbon atom and 1 phosphate atom, and another similar molecule called dihydroacetone phosphate. It also contains 3 carbon atom and a phosphate atom. However, this dihydroacetone phosphate is unstable and it immediately turns itself into glyceraldehyde 3-phosphate. So we started with fructose 1,6-bisphosphate and now we are ending with 2 molecules of glyceraldehyde 3-phosphate. From here onwards, both glyceraldehyde 3-phosphate will follow the same pathway. That's why we are not going to study them individually, rather we are going to just stick to one of these glyceraldehyde 3-phosphate. To these 3 carbon compound, which already has a phosphate group attached to it, another phosphate group is added, and this makes it 1,3-bisphosphoglycerate. So now you can see 3 carbons with 2 phosphate groups attached to it. I know it looks like poop, which this topic actually kind of is. Anyways, moving on. The 1,3-bisphosphoglycerate eventually lose one of these phosphate atom, and it becomes 3-phosphoglycerate. So now we are again back to the 3 carbon compound attached to 1 phosphate atom. Then the 3-phosphoglycerate converts into phosphoenol pyruvate, which still is a 3 carbon compound with a phosphate group attached to it. And then finally, the phosphoenol pyruvate decides to lose the last phosphate atom and converts itself into the 3 carbon compound called the pyruvate. And so now we are left with this carbon 3 compound without any phosphate. So now that we understand how the glucose ends up being a pyruvate molecule, it's now time to understand what happens to the phosphates. Well, at this point, as you can see, the 6 carbon compound is suddenly turning into a 6 carbon compound added to a phosphate group. So where does this phosphate comes from? You have guessed it right. It comes from a molecule of ATP. So a molecule of ATP enters and goes out the ADP. And the phosphate that was lost by the ATP has been joined or tagged in to this glucose 6-phosphate. Here again, we can see that the 6 carbon compound is receiving another phosphate to become a bisphosphate. So where does this phosphate come from? Again, it comes from another ATP. Then the fructose 1,6-bisphosphate split to form 2-glyceraldehyde 3-phosphate. Here the number of carbon and the number of phosphate are still balanced. Over here, however, some changes do occur. As you can see that the glyceraldehyde 3-phosphate becomes another bisphosphate. So somewhere in the line, a phosphate has been added to the glyceraldehyde 3-phosphate. So where does this phosphate come from? Does it come from another ATP? Unfortunately, no. Rather, this phosphate is brought by a molecule of NAD. So this time there will be a NAD carrying a phosphate group, and after adding the phosphate, the NAD will be converted into a molecule of NADH. Remember that there are 2-glyceraldehyde 3-phosphate, so actually 2 phosphate groups will be added to each of them. Anyways, at the end of it, you will be left with 1,3-bisphosphoglycerate, a 3-carbon compound with 2 phosphate groups attached to it. At this point, you will notice that the bisphosphate is getting converted into a 3-phosphoglycerate. That means a molecule of phosphate is being lost. And where is it going? Of course, it's going to form a molecule of ATP. So, and ADP comes in and ATP goes out. Since there are 2 1,3-bisphosphoglycerate, so at the end of this cycle there will be 2 ATP production. Then there is not much change over here. The number of carbon and phosphate are remaining the same. However, again, as the phosphoenol pyruvate gets converted into pyruvate, another phosphate molecule is lost. So it again forms another molecule of ATP. So another ADP comes in and goes out as an ATP. And the phosphate is lost, and we end up with the pyruvate. And now for the enzymes. Now this seems to be a bit tricky to remember the name of all these enzymes, but if you try to understand these names, then the task of memorizing becomes much more easier. Let's try to understand the first step. In the first step, the glucose molecule, which is a hexose sugar, is getting added to a phosphate group. Now, any enzyme which helps to add a phosphate group is known as a kinase. And since this kinase is working on a glucose molecule, so this first enzyme is known as a hexokinase. Next. Now, this phosphorylated glucose changes its shapes or becomes an isomer of itself and becomes fructose 6-phosphate. Understand, nothing has been added, only a phosphorylated glucose has been isomerized. That's why the name of this enzyme is known as phosphoglucose isomerase. So PGI. See, all the name of the enzymes are actually related to their functions. So to remember the name of the enzymes, you just need to remember what is actually happening. So now what is happening? A phosphate molecule is being added to a fructose molecule. So what will be the name of this enzyme? Phosphofructokinase. So phosphofructokinase. Now this one is a bit tricky because fructose 1,6-bisphosphate is being broken down into glyceraldehyde 3-phosphate, and the name of the enzyme is aldolase. Now the problem is the name aldolase doesn't actually relate to what is going on over here. However, you have to understand, where does the word aldo come from? Aldo in Italian, it means tall, and as you can see that the molecule fructose 1,6-bisphosphate, it is quite a tall molecule and it is being broken down. So aldo, the tall molecule, and lysis or splitting. So since the tall molecule is broken down, so we can remember it like this: aldolase, the enzyme that breaks the tall molecule into shorter one. Next, the glyceraldehyde 3-phosphate is being converted into 1,3-bisphosphoglycerate. And as we have seen earlier, this happens with the help of an NAD. And at the end of the process, the NAD becomes NADH. So where is this hydrogen coming from? It is coming from the glyceraldehyde 3-phosphate. So in this process, the glyceraldehyde 3-phosphate is losing hydrogen. And the enzyme catalyzing this reaction, the name is quite straightforward. Glyceraldehyde 3-phosphate dehydrogenase. Next, 1,3-bisphosphoglycerate is becoming 3-phosphoglycerate. What is happening here? This glycerate molecule is losing a phosphate. So since there is movement of phosphate, it is definitely going to have the name kinase. And since this is a phosphoglycerate, so the name is phosphoglycerate kinase. Next, the 3-phosphoglycerate becomes converted into 2-phosphoglycerate. Now, although I haven't written this step in the board, you can understand it like this. 3-phosphoglycerate becoming 2-phosphoglycerate. So basically the phosphate atom is jumping the carbon atoms, and this occurs with the help of an enzyme called phosphoglycerate mutase. Then the 2-phosphoglycerate gets converted into phosphoenol pyruvate, and the name over here is quite straightforward. The name of this enzyme is enolase. And lastly, the phosphoenol pyruvate is releasing a phosphate atom, so again there will be kinase, and since there is pyruvate, so the name of this final enzyme is pyruvate kinase. So that fixed the enzymes. Okay, I know what you're thinking right now. So we understand what happens, how it happens, but we cannot memorize this process. Actually, there is a trick of how to memorize these type of complicated diagrams. You have to start by asking small questions. Questions such as, number one, how many different type of molecules are involved in this reaction? So let us start. So first it starts with glucose. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So how many molecules? 10 molecules. Next you have to understand how many steps are involved. So again, from here to here, one step. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 steps. Now this also answers another question. Since there are 9 steps, so there must be 9 enzymes. Now let us bring this reaction down into some major stages. So what are the major stages? First, the glucose, through a series of reactions, become this molecule, the fructose 1,6-bisphosphate, which is the largest molecule in the entire reaction. So this is the first molecule, and this is the fourth molecule. And then the next major step is when this large molecule splits exactly in half and results in glyceraldehyde 3-phosphate. And this is the fifth molecule. The next major step, I think, is the ultimate conversion of this glyceraldehyde 3-phosphate into the final pyruvate molecule. And this is the 10th molecule. Now it is very easy to remember these 4 names, which is the glucose, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, and finally the pyruvate. So we have already memorized 4 names out of 10. My analysis is that if you can remember the name glucose and fructose 1,6-bisphosphate, you will also be easily able to remember these 2 names. So that gets down with number 2 and number 3. So now we have memorized 1, 2, 3, 4, 5 and 6 molecules. Only 4 more to go. Now pyruvate comes from another pyruvate, which is phosphoenol pyruvate. So we have also memorized the name of number 9. 3 more to go. Out of these 3, there are 2 phosphoglycerates. One is 3-phosphoglycerate and the other one is 2-phosphoglycerate. So that gets down with number 8 and number 7. And the last one is another bisphosphoglycerate. We have already studied another bisphosphoglycerate at number 4. So number 4 and number 6 are similar. So that's how you can memorize the name of all the 10 compounds that are associated with glycolysis. Now the challenge of remembering where phosphates are added and removed. This will also answer where the kinases are involved. So here's the trick. Just remember this. The first transaction is occurring between 1 and 2 molecule, the first and second molecule. And then between third and fourth molecule. Again between 6th and 7th molecule. And finally between 9th and 10th molecule. So how can you remember it? Very simple. 1, 2, 3, 4. Like the children remembers the number 1, 2, 3, 4. And then the next line 6, 7. And let's turn the 6 upright. 9. And what comes after 9? 10. So here are the phosphate reactions or the kinase reactions, which is 1, 2, 3, 4, 6, 7. Invert the 6, 9, 10. So I hope that is going to help you to understand and remember glycolysis. If you have any other questions or any other problems in any other topic, please let me know in the comment. Thank you.